Yeah, you're going to have to tell me how to get rid of that. Oh.
You know, I found it interesting that they have found fossils, marine fossils in high elevations.
Oh, yeah.
Evidence of a global flood.
Yeah. How else would they be there? Well, there could be uplift, I guess, but yeah. Yeah. Lots of that going on, yeah.
We don't hear too much about it, though.
No, no. There's a lot we don't hear when it's not with the supposed paradigm they've got going. So, yeah, we won't hear it. Am I ready? Okay, I'm ready if y'all are ready. Thank y'all for staying. This is really good. Y'all got through 2 hours and you're back again. That's really nice. Yeah. This is the part I really wanted to talk about, and I really like talking about this because biology is starting to show so much about God, and I think you should enjoy this a lot. It's really close to me here, so let's go in there. We're going to revisit evolution a little bit and look at the science for creation. When I started teaching the course Evolution, there was really only one book that I found that was on the creation side, and it was Michael Denton's book, Evolution, A Theory in Crisis. Well, it's interesting. He wrote it a second time in 2016. It's Evolution, Still a Theory in Crisis. It just seems like the more and more evidence that we come up across and more things we find out in science, it just always confirms there is a God. And so that's just really kind of fun. One of the guys who really influenced me a lot when I first started teaching evolution was Michael Behe. He was a biochemist. He still is a biochemist at Lehigh University. And he works with the bacteria. He wrote this book, Darwin's Black Box. The story behind that is he worked with the bacteria. And he was looking at the bacteria flagella, which y'all can see the flagella here on the end of the bacteria, and how it's attached to the membrane is this motor. He calls it a motor. And you can see here, he says it's got a bushing on it, it's got a drive shaft, it's got a rotor, it's got a stator. It's made up of about 40 proteins, and it rotates in there. And he said, that looks like an outdoor motor. So he called this, and other systems that he looks at, irreducibly complex machines. That is a machine that's composed of several well-matched matching parts, interacting parts that contribute to the basic function. If you remove any one of those parts, it doesn't work. And so that means all those parts had to be there right there at the beginning for that thing to work. So he searched the literature to find out how this machine, this motor for this flagella, how it came about by gradual evolution. Searched the literature, found nothing. Found nothing. So he wrote the book, and I'll go back to it, Darwin's Black Box, and turned the biology world upside down, because at the end of the book he said, this had to be designed. This is like a motor, it's just had to be designed some way or other. And so he has continued to answer all of the criticism he's received. He answers it back in a scientific way with scientific evidence, and he's now in his third book, which I'll tell you about here in just a minute. So he just does a really good job looking at these machines. One of them is clotting blood. In order for us to clot our blood, it takes all of those proteins, all of these proteins in here to clot blood. I can't use my left hand. If you get yourself cut here, then you have a factor in your blood called Hageman factor. It changes it to Hageman prime, and then that changes that one to that one, and that one to that one, and that one to that, and that, and all the way down to clotting blood. And all those parts have to be in place. And he says this is a irreducibly complex system. You can't remove any one part, or your blood won't clot. And so you find these machines everywhere in your body. Now then, I'm going to switch over to evolution. Here's evolution's story. Basically, this is their story. 3 parts to it. Number 1, there are mutations. Some are good, some are bad. And then they say natural selection occurs. There's competition in nature, and certain mutations will be selected over others, and so that occurs. And then, number 3, selection. If you repeat it over and over again, it's going to lead up to a new species, and in the process, build up all these wonderful structures of life. That's evolution's, that's their story. That's their story. Well, number 1, yes, there are mutations. There are mutations. But it's really not what you think. They say some are good, some are bad. So I'm going to ask you, for every one beneficial mutation, how many harmful ones do you think there are? What would you guess? Anybody want to make a guess? 4. I'm sorry? 4. Okay, I got 4. Anybody else want to guess? 200? Okay. It's interesting. They don't tell us this. Did y'all know that? Nobody ever hears this. There is a population geneticist who's probably the most famous population geneticist in the world. He retired. J.C. Sanford is his name, worked at Cornell University. And when he retired, he wrote a book. And he said, we have been looking for evidence for evolution for a long time. Can't find any. We can't find any. And in his book, he mentioned this. He said, they'll never tell you this. For every one beneficial mutation, are y'all ready? There's 1 million harmful ones. 1 million harmful ones. So the chance of getting any mutation that'll be beneficial and even pass it on and fix it into the population is just quite low. And so they don't tell you that. They won't tell you that in the textbooks. Number 2, natural selection. Yeah, natural selection occurs. We do see within a species, we do see if this particular form of a species is better adapted to that environment, it will take over the environment because it lives better in that particular environment there. So we do see that, but it stays within that genus species. It never causes an organism to gain a structure or trait. It can't create a trait. All it can do is select against a trait. That's all natural selection can do. But it seems to do wonders in the evolution world that we haven't figured out how that has happened at all. And so, yeah, that's natural selection. It does occur, but all it can do is select against something. That's all it can do. Then this last point, selection repeated over and over again. We've never seen that. That's the one thing they add on that there's no evidence for that. They have to say mutations have accumulated over time because we went from a bacteria to all the different organisms we have today. So somewhere along the way they had to accumulate, but there's no way for that to happen. We never have seen that in biology. There's no mechanism for that to happen. We've never seen any new kind of structure form from anything. And so they've got a couple of things that are true. There is mutation in natural selection, but it doesn't work like they want it to. And that last part just doesn't happen. It just doesn't happen at all. So what we're going to do is look at some mutations and some organisms and help you understand what's going on with all of those. So I like the polar bear. Y'all like the polar bear? I do too. It supposedly evolved from the brown bear. And if I had to answer that question, I would say, yeah, it probably evolved from the brown bear. They were considered the same species until 1774, and then they were made two different species. They're the same genus, but they're two different species. But it kind of makes sense to me. One of them looks very different, and it lives in a completely different environment from the brown bear. So I'm okay with making them two different species. So you can see there on the bottom, they're in the same genus. They're in the same genus here. They're in the bear family. But they're quite different, and so they're made in two different species. And I'm showing you that chart because that's how we do taxonomy, and we put things in different categories. We've never seen anything jump from one family to another family. We never see that. All the dogs, they can interbreed with each other, but you never see them jump into another family, a cat family or anything like that. So basically we're thinking family is the same as kind. When God took two of every kind on the ark, that's probably family. So he took two dogs, maybe two wolves or something, and from that we get all the different dogs that we see today. And so we do see variation. We do see different forms of traits occur, but they always stay in that family. Does that make sense? Yeah. So the whale family, we've got dolphins and whales in the whale family, and dolphins and whales can interbreed with each other and produce viable offspring. They've done that in the San Diego Zoo. So that's that kind. Does that make sense? Yeah. So when you talk family, that's the kind that we're talking about in the Bible. Well, the polar bear, lighter in color, more resistant to cold, more adapted to eating seal blubber. So each one of those characters has an advantage over previous steps of evolution for that organism in the Arctic region up north. So it's a beneficial mutations there. Well, we can do the genetics. In the genetics, there's two genes that were affected. One was the APOB gene, making the bear more likely to eat fat. So it really changed the LDL part of cholesterol there to where it wouldn't carry cholesterol, so it doesn't have to worry about heart attacks. It can eat all the seal blubber it wants to eat and doesn't have to worry about it. What happened to that gene was it was degraded, or its function was destroyed in that gene. It broke. The gene broke. The other gene, LYST, associated with coat color, it changed the gene to where it couldn't make pigment. And so it was a damaged gene, or a damaging gene that happened there. So the way we got the polar bear was not by adding any new genetic material. All it did was break a couple of genes, and we got the polar bear. Does that make sense? That's what we're seeing everywhere. Every type of evidence they use for evolution, it's a broken gene. It's a broken gene. So the polar bear adapted not by adding new genes, but by breaking a gene. So we see the same thing in humans. The sickle cell gene, that's just a single base change in the DNA, and it makes the red blood cell sickle shape. But in Africa, you're resistant to malaria. Beneficial mutation, but it's a broken gene. The OCA2 gene, a mutation in that gene caused the loss of ability to produce pigment, the melanin in the eye, so you have blue eyes. Beneficial, a lot of people like blue eyes, but it's a broken gene. You see the point? Yeah. So it's with our new technology and genetics, we can now look at this and test Darwin's theory. And most of these beneficial mutations are broken genes. So it's just kind of fun. That confirms Genesis 2. Not Genesis 1 in particular, but Genesis 2, the first few verses. Listen to what they say. Thus the heavens and the earth were completed, and all of their host. By the seventh day, God completed his work, which he had done, and he rested on the seventh day from all the work that he had done. Then God blessed the seventh day and sanctified it, because in it he rested from his work which God had created and made. God created all the DNA for every organism all in those 6 days of creation. Yeah, things break. We get new traits from that. Not a new structure, but a different form of a trait in some things, but we stay the same kind in all that. Does that make sense? Yeah.
So things are being broken down.
Yeah. Yeah. Kinda. I'll explain that here in just a minute. At this point in biology, we were saying it's broken genes. It is just broken genes. But the last couple of years, I'll show you what we're finding here in just a second. That's why I wanted y'all to really hear this, because it's really fun. It's really fun. So, but yeah, a lot of the genes are broken, and well, when we get to the dogs, that'll become quite clear. So, we see Darwinian evolution proceeding mainly by damaging or breaking genes. So, it will work for small-scale adaptation, change at the genus-species level. We might get a less showy guppy, so it won't be eaten by the predators as bad. And so we do see those kind of changes, but not at the level of the family, not at that level at all. So there we go. There's Behe's third book. It's called Darwin Devolves. It came out in 2019, just a really good book. He kind of explores how all of these evidences for evolution really are just showing broken genes. Since 2019, we found out more, and I'll try to explain that here in just a minute. All of Darwin's finches, where Darwin got his idea about evolution, there's about 12 to 14 different species of finches on the Galapagos Islands over there, different colors, different beak sizes. He divides them up kind of into 4 groups based on where they live, what they eat, other characteristics. There they are. You can see the different finches. He had just been over to South America and then traveled over to the Galapagos Islands, which is closest to South America. He saw the finches over here, and he noticed these are real similar, but they do have some different characters, different colors, different beak size, and those kind of things. And so there you can see he traveled. It's not far from South America here, where he saw the finches, over to the Galapagos Islands here. And so that's where he got his idea. These birds got blown over here, they can change. And he just thought, if it can just keep on changing, maybe it can change into something else. And so that's where he got his idea of evolution. Now then, these finches probably did get blown over to the Galapagos Islands. They're volcanic islands, so they have lots of different niches. And some of these traits better fit this environment. Some of the traits better fit this environment. And so they kind of become separated. And certain islands had different birds on them. So, yeah, it is a little bit of adaptation there. Peter and Rosemary Grant were 2 scientists who spent their life, really, on the Galapagos Islands, just studying the finches on this one island, Daphne Major. And they documented everything about all the birds on that island. They knew what bird mated with what bird. They knew what color it was, its beak size. They measured everything. You can see this came from a textbook. They looked at beak size, and this is in the evolution chapter in the books. They noticed beak size in 1976 was 8.8 millimeters, the average beak size. But then they had a drought in 1977, and in 1978, the beak size went up to 9.8. So the technical definition of evolution is, it is a change in a trait over generations. So we did see beak size got larger. So what happened on the island? There was a drought. So all the small plants, which did not have very deep roots, they died. The larger plants, which had deep roots, were able to survive the drought, but they have larger seeds. So the only birds that had a big enough beak to break the seeds survived. All the smaller birds died off. So average beak size got larger. Does that make sense? Yeah, it just happens. So that's what's given in the textbook. It is a change in a trait over generations. So I looked at that, and I decided to go look at their original data that they published back in Nature, and sure enough, that's what happened. So here's beak size. It did go up in 1978, but then it came back down. It rained. And then it went up, and then it went way down on beak size and stayed down for a long time. Then it got droughty again and went back up and then down. So over time, the beak size just goes kind of up and down. And what happened was other birds from other islands flew back onto that island and reduced the beak size down when it rained. So it's just, that's what's going on with all of that. It's really not evolution. Things really doesn't change very much over time. So they actually kept the DNA for these birds. They looked at the DNA of these and actually analyzed them here recently. And what they found was they looked at master genes on the DNA for head and beak development. And one of the genes, ALXI, that affected whether it was blunt beak or pointed beak, just that one gene. And so a mutation in that one gene would just make a little bit difference in the beaks. 2 mutations in that gene resulted in differences between blunt and pointed beaks. All of their differences were just like that. Just a little change in a gene here and there, mainly breaking the gene that caused all of those changes. The cichlid fish in all the lakes over in Africa, all kinds of different colors and stripes on these and shapes of the fish. They say it's just a world of evolution occurring over there, 400 or 500 different species of these fish. Well, if you look at the genetics of these, and they have looked at the genetics in this lake and in this lake and in this lake, they're looking at the genetics of all those. One gene that EDNRB1, the mutant gene had changes in 2 regions. 2 traits controlled by the normal protein were lost by the mutant. So color and stripes were lost. And so it's just broken genes that are causing most of those traits. And what's really interesting is, if you look at this lake over here, it has evolved into several different forms of cichlid fish. And it's the exact same forms that evolved in this lake. And it's the exact same forms that evolved in this lake. So the ancestors for all 3 lakes had the same DNA, and they evolved the same because there were similar environments in each of those 3 lakes. They kind of changed the same. So it's like there was the same original DNA, but it just changed in a few different ways. Sometimes breaking genes, mostly by breaking genes, but also by jumping genes. Genes jump in the DNA to another chromosome. It's really kind of crazy how that works. I'll talk more about that here in just a minute. Yeah.
Does that mutation gene become dominant?
Does it become dominant? Some can be dominant. Usually the normal gene will be the dominant one, and when it breaks, then we get the other form, which then we would consider it recessive and not the dominant trait. But it really depends upon the gene that we're looking at.
What I'm referring to, does reproduction of that dominant gene or that defective mutated gene become
Fixed in the population or something like that? Yeah, it usually does. And that's the one thing that Darwin didn't understand is, yeah, we had mutations in those birds that flew over from South America or got blown over, but once they get into a certain environment, most organisms, they go to, for each gene, there could be a dominant and a recessive, they go to one or the other. So in this particular environment, the dominant gene may be favored. In this environment over here, maybe the recessive gene would be favored. But they all go to homozygous condition to where it's only one form. So they can't change anymore. And that's what's going on. Darwin thought they could keep on changing and become something else, but they were at the end of the road. they had changed all they really could to fit that environment. It's like the polar bear. The polar bear, once it got those traits to live in the Arctic region, it's pretty much limited to that Arctic region. It can't change anymore. And so that's what we're finding is, sometimes they're recessive, sometimes they're dominant. And they usually take over the population. Because any population tends to go to homozygosity, to where both genes that you have are the same. They tend to do that. I don't know if that makes sense. I'm getting too genetic-y here, I know, yeah.
Basically, everything is environmentally driven in genetics.
I think we could say
In
that.
terms of changes for them to
Yeah.
adapt
Yes, yes,
to an area.
yes.
Because we know, and I say we know, I mean, you tell me, we know that there are animals that have gone extinct,
Yes,
that are no longer with us.
right.
They just didn't adapt.
That's exactly right.
And somehow, man is to blame for all of that. You know?
Yeah.
And I'm not saying we're not responsible in some ways.
Yeah.
I'm just saying that, nature has a way.
Yes, yes. Well, when everything came off the ark, it had to be distributed throughout all the world at that particular time. They had to go to all places in the world. And so God built in to our DNA the ability to adapt to different environments. And so if these organisms went this way, they would adapt to that environment. If they went this way, they would adapt to that environment. So if it was a camel, if they went this way, they adapted to that environment, but they stayed a camel. If they went this way, they stayed a camel, but they adapted to that environment. If they went to this environment, they just couldn't survive, they died. Yeah, they stayed a camel. Does that make sense? Yeah, that's what's going on. It's just incredible how much is built into our DNA that we just didn't realize. And now that we can sequence genomes, it's just become amazing. And I'll show you that here in just a second. Yeah. These are good questions. These are really good. Lindsay.
Photographic evidence is something too.
Photographic evidence.
I'm not a good example, but pictures of my ancestors, they're much smaller than I am.
Yeah, that's exactly right. So we have grown several inches over the last few years. The average male has grown several inches. Why do you think so? Probably diet, maybe pharmaceuticals and medication probably helped that along a little bit too. We change a little bit over time. We're still human though. We're still in that kind. Linsky.
Does this
Go ahead.
affect our pigmentation?
I'm sorry?
Does it affect our pigmentation?
Yes.
Different color of people?
Yeah, we have all different colors. Yeah, and that effect. If I moved to the equator, I would get a little darker. I would start producing more melanin, and I would get darker. I move to the north, I'll probably get lighter, because I don't have much. There's not that much sunlight up there, so I'd probably get lighter in color. So that affects us. Different pigmentations. We have two types of melanin, eumelanin, pheomelanin. So we have those differences, and by sexual recombination, we get different combinations. My wife is more of the yellow color. I'm more of the red color. And so our children have different variations of that. So yeah. I'm sorry? Yeah, and the black color, lots of melanin produced in that. So I don't know how that came to be. Well, I'll get to that in just a minute, okay? Yeah. We have mechanisms in our DNA that is almost unbelievable, that's built in, that God put in there so that we could adapt to most environments. It's just amazing how it works. And it usually involves genes that jump from one chromosome into another chromosome. And when it jumps into not a random place, but a hot spot where it usually jumps. That God designed us that way so that we can handle change and stress. So it's incredible how we can change. I know that sounds sci-fi, but that's what's going on. I'll show you in just a minute something on that. This is Lemsky's experiment on bacteria. This is single-cell bacteria. He ran this for 25 years. He took 12 flasks of E. coli, same E. coli in all 12 flasks. He just let them multiply. So E. coli multiply every 20 minutes, and so you can get 1 million E. coli in one day from one bacteria. And so every day, he had graduate students that would take out 1% of the population and put it in another flask. And so every day, he did this for 25 years. They just went from generation to generation to generation, 25 years. Long time. 65,000 generations. That's equivalent to over 1 million years in the history of a large animal. So there should be some kind of evolution in these populations. There was. In all 12 lines, there was evolution. After 1 month, they started growing faster. After 2,000 generations, the growth rate was 37% faster. Remarkable growth. And in all 12 lines. And after 50,000 generations, the growth rate was 70% faster. They were growing faster. So something was changing in that E. coli bacteria. Something was changing. So they also got fatter. They were 85% increase in volume. So they got fatter, they were growing faster. Evolution was definitely occurring in all these lines, all 12 lines. They came in different ways. This one evolved quicker than this one over here, but they were evolving as they went along the way. Well, they looked at the DNA. They kept DNA after so many generations. They would freeze that bacteria so they'd have the DNA to look at it. So the first helpful mutation, the cells lost the ability to metabolize ribose. So they couldn't break down ribose for energy. All 12 lines suffered the same deletion. But they started growing faster because they figured out a different way to use a different sugar to metabolize. And so they actually did better and started growing faster and even getting bigger. So more mutations were tracked. All of them were broken genes that were helpful in yielding faster growing cells. 59 genes had changed activity, either increasing or decreasing it, mostly decreasing the activity, in 8 of the 12 lines. So it's just a few mutations in genes, but they had beneficial mutations. We actually saw it evolve. And so all the beneficial mutations were by degrading or outright breaking the respective ancestor gene. So it's not making new genes or new genetic material, it's just breaking a gene or inserting something into a gene to where it wouldn't function correctly. And that's usually what happened. So there's 1,000 ways to break a gene, but just a few to improve it. So it's easier to get a damaging mutation. And that's usually what happens. So they've done lots of experiments on these kind of things. This experiment, it just came out in the last couple of years. It's with bacteria, and I want to just kind of explain how this works a little bit. And I'm sorry this is too sciencey, but it's really kind of interesting. So they took these bacteria and they starved them. They put them in a media that did not have any sugars that they would be able to eat or take into their body and metabolize for energy. And so the only thing they put in the media was beta-glucosidase. And we did not know of any bacteria that could metabolize beta-glucosidase. And so I was listening to a guy talk about this, and he said, I hate to say the word beta-glucosidase all the time, so he put the initials up there, BGL for beta-glucosidase, and he called it bagel. He said, it's a lot easier to say bagel, so if I say bagel, that's why I mean that sugar. So they knew this bacteria could not metabolize that. So they let the bacteria go for 7 days. That's a long time for bacteria because they reproduce every 20 minutes. And so they had millions of offspring, but they were starving and just barely existing. Then after 7 days, this one bacteria just started reproducing like crazy. It started metabolizing beta-glucosidase. And it just started taking over the population, and the whole population was just growing just fine. And if we just stopped right there, we would say it evolved. It evolved a new gene to break down this sugar. And that's the way evolution has always worked in all this. Well, with our technology, they were able to find out exactly what was going on. It so happened in the bacteria DNA, so this is the bacterial DNA right here. In that DNA, there were 3 genes right together. Now, in bacteria, they're a little different than we are. If we have a gene on our DNA, right next to it we have a promoter, and that promoter, a protein binds to it to make that gene so that we can make a protein out of it. So we need a promoter for every gene to make it work. Does that make sense? Okay. Well, in bacteria, they can have 3 genes together with only 1 promoter. Here's the promoter over here. And so it could turn on all 3 genes at the same time. And it's really interesting. What they found was, here's the gene that could metabolize the bagel. The other gene here was a protein that was made that went into the membrane of the bacteria that was able to sense if there's beta-glucosidase in the environment. So it was a sensing protein to let the bacteria know it's here in the environment. And the third gene, the one in the middle here, that was a gene that went into the membrane that would pull the beta-glucosidase into the cell. So you've got one gene that senses it in the environment, another gene pulls it into the cell, and then another gene metabolizes it. Isn't that incredible? This DNA was already there in the bacterial DNA. The genes were there to utilize beta-glucosidase, but it was shut down because it's got this little thing right here. It's called a SID. That's a stress-induced duplex destabilization place. It's where the DNA becomes so tightly wound that it forms a loop in there, or a big open spot. And then this part of the DNA will come down and attach to it. I'll show you the next slide. It'll come down, and this protein, HNS, attaches to it, and it shuts down the DNA here so that this promoter can't turn those genes on. So it's a built-in mechanism to not turn those genes on because usually you don't meet up with beta-glucosidase. But if there's beta-glucosidase in the environment and the organism is stressed, then the stress, when a bacteria is stressed, it releases tons of this other compound called cyclic DNA, cyclic AMP. And when it releases the cyclic AMP, that opened up that block right there, and that allows these 3 genes to be made in a small amount. And when they start bringing in the beta-glucosidase into the cell, then that triggers another gene to be turned on. That's a jumping gene. It jumps into the DNA right here. There you go. It jumps into there and makes it a permanent connection so that from now on, this organism can use beta-glucosidase. So what I'm saying is, I know that's complicated, but evolution would just say it, and it's always said in the past, oh, it got a new gene. It mutated. It's a new gene. But in actuality, we're finding the genes are already there. They're built in. We have a or the bacteria has a mechanism to keep those genes shut down because they're not needed and they don't expenditure any energy on those genes. But then when it comes time for stress and this other thing shows up, they've got the DNA to do it, and they've even got a mechanism to turn it on and off whenever they need it. Do y'all get my point? We are designed very well. And a lot of genes in organisms are turned off, but they're there in case they meet an environment where they need the genes, and then God's got a way to make them turn on. And a lot of it's by a gene that jumps. Is that not incredible? You and I, they've been keeping track of how many genes we've got. We've got 63,300 genes right now. That's what we know of. That number keeps increasing every year. That's a lot of genes, right? Is 63,000 a lot of genes? Guess how many jump? I'm talking jump from one chromosome into another chromosome, jumping from here over into here to maybe jump in as a promoter to make another gene work that's been turned off. Half of our genes are like that. Half of our genes are like that. Most of them they're finding out work as our brain develops in the embryo, as a baby develops in the embryo, the brain starts developing in a certain way, and then a bunch of genes jump, and that makes it start developing this way, and then they jump, and the others jump and make it develop a certain way, and our development depends upon that. We're designed to be made that way. Isn't that incredible? 10 years ago, I wouldn't have believed myself saying that, but we have these jumping genes, and they tend to turn on when we're stressed. If we get in really hard stress, our body starts having genes jump to see if one will jump into a position that will turn something on that can get us through the stress. That's just incredible, just incredible.
I'm not sure this connects, but I've just recently read an article where people that are paralyzed, and that have broken vertebrae
Right.
back,
Uh-huh.
they've introduced, and it's been successful, a peptide that has bridged these damaged. Is that connected to our DNA?
I don't know how to answer that. I know our DNA has codes for every peptide that is made, for every protein that's made. And so I don't know what they're inserting. Are they inserting something into the DNA, or are they inserting the protein that causes the DNA to do something? I don't know.
All I know is that I read it is a peptide that's introduced by doctors and
Okay,
scientists
yeah,
that they've developed and put into, the patient,
yeah,
and then they're walking.
yeah,
I mean, these people are regenerating,
yeah.
getting to the point to where they walk.
That's amazing.
It is.
It is. Yeah. I'm telling you, we've got mechanisms.
The complexity of it.
That's it. God built in lots of stuff there, and it's a lot more complicated than what we think. And so even when I'm saying, okay, this was a broken gene, I'm not really sure it was a broken gene that was not intended to be broken. Does that make sense? And if you look at this, let me get away from this guy. There's a peppered moth. We'll talk about that later. Yeah, the woolly mammoth. The woolly mammoth, we've sequenced its genome. And what they found is, it's just like the other elephants, just exactly like them, except there were 87 genes that had deletions. So you can see down there on the DNA of the woolly mammoth, it's gone. The yellow gene is gone. It just fell out. It got broken out. Or they were indels. An indel is an insertion, that's the end part, an insertion that deletes the function of the gene because it inserted and broke it up. And so 87 breakages, that's the difference. But it made the woolly mammoth adapt to its environment that it was in. And it could live in that environment, kind of like the polar bear could live in its environment. And so those are mechanisms that were built in to that organism to live in a certain environment. It's just amazing.
You brought this up, and I do a lot of reading.
Okay, yeah. Yeah.
They're talking like they can reconstitute the woolly mammoth.
Oh, yeah. And it's just a matter.
Bring it back.
Yeah, they might be able to, but it's kind of complicated on how genes jump. God's got that part figured out, and he knows how he made it to make them jump. You and I can't make my genes jump. Now, if I get in stress a lot, we have seen it in humans where genes jump in stress. But can we make the right genes jump in the place we want them to? I doubt it. I doubt it. But I'm just saying, God designed us to do this. And all the evidences of evolution where they're showing changes, it's really showing the built-in complexity that our DNA has. It's just amazing. Does that make sense to everybody? Kinda? I'm a geneticist. I love this stuff. Yeah, sorry. It's just that way. Other genes, some people are resistant to developing diabetes. They'll just never be able to develop diabetes. It's a broken gene. Some people never have a heart attack. And that's because that gene APOC3 lowers your cholesterol levels. It's the same gene that the polar bear had. And so there we go. It's just there. So, yeah. The dogs, we're getting new breeds of dogs all the time. Do you know how they get a new breed? Broken genes. And all the new breeds tend to have to go to the vet more often because of all the broken genes that's there. And so it really makes it hard to keep those animals alive, but they can keep them alive, but they have a new trait. So just interesting. If you look at the different traits, increased muscle mass in some breeds of dogs from degrading a myostatin gene. Yellow coat color, loss of function of melanocortin. Black coat color, deletion of a glycine residue from the beta defensin protein. So they're all broken genes. It's just kind of fun. Size, there's 6 different genes that control size. 3 of the genes are degrading changes in the protein, and the other 3 were on the control regions of the genes. My wife, she has a genetic disorder. Her red blood cells are not made right. Your red blood cells have 2 alpha proteins and 2 beta proteins to make your hemoglobin. Her beta is not made right. She's got the gene to make the beta, but she doesn't have the promoter to make the gene. So just different things that are there. She does just fine. She's tired all the time, but she does just fine. Short muzzle, it's associated with mutations in those 2 genes, lessening their activity, and a joint mutation in BMP3 that damages the protein. So lots of things. Some of these genes, though, can occur by jumping genes. And that's what we don't know. We used to just say they're broken genes, but now we're kind of questioning things. Even the friendliness of dogs towards humans compared to a wolf, it's associated with the disruption of the genes GTF21 and G2F21RD1. And if you disrupt those genes in humans, that leads to outgoing personality plus mental disability. So we share a lot of genes. We do with other organisms. We share a lot of genes in all of that. So that's evolution's greatest problem. Almost all beneficial mutations are broken genes. The more genes that are degraded for short-term evolutionary adaptation, the fewer are available for future adaptation. Once that polar bear has those broken genes, it's pretty much limited to just that area. Natural selection only works at the genus species level. No evidence of new genetic material ever being made. So no evidence for the evolution of simple forms of life to the more complex. So it's just kind of fun. God made all of our DNA, and he made it very complex, and it helps us adapt, and that's what's going on. Okay, so is there any evidence for creation? I'm going to hurry here through this. This is really fun. There's evidence for creation. This came out in Science magazine. There's an insect that jumps, and he jumps with gears on his leg. There they are. It's got gears. So its two legs, its hind legs, had these gears hooking the two legs together. And then when he cocks his legs to jump, then when he jumps, the gears keep his legs moving the same so that he can jump exactly where he wants to go. Is that not incredible? Just fun. There's a nymph, and you can see the gears are starting to develop on the nymph and how they're attached to the legs there. And so that way it can jump accurately. Just really, really kind of fun. The National Geographic writer, when they had an article about that, said, this insect has gears, gears. He's kind of surprised because where do you see gears? In an engineering shop. Yeah, you don't see them on an insect. So that's just really, really kind of fun. In your eye, we now know there's Mueller cells in our eye. Now, I made a little comment. This was found in the Proceedings of the National Academy of Sciences. You can see that. But my note there at the bottom, the eye's always been amazing in how it operates. But to me, it's equally amazing how every time in a textbook it talks about the eye, it talks about how imperfect it is. And they'll say it's imperfect because of two things. It's got a blind spot. You have a blind spot in your eye. And all of the photoreceptors that detect the light. So the light comes in, an image forms back here on the retina, on that orange part. And so it's going to appear right here. But all the rod and cone cells are way back here. And they will tell you in the textbook, if this was created by a God, he should have put the rod and cone cells here to have maximum absorption. They even say that in the textbooks now, that this is an imperfect eye. It's not made right. Well, they didn't know about Mueller cells. They found out, now we have these Mueller cells. These are cells that when light hits the retina right here, they take the light, they're called wavelength-dependent waveguides. They'll take the green and the red light to certain cones down here, and they'll take the blue and the purple to adjacent rods. So these Mueller cells are actually taking the light, dividing it up, and putting it into its proper cell down at the back. And it needs that distance for that to occur, for it to separate it. It increases photon absorption by the cone cells. Oh, I didn't mean to do that. I'm hitting the wrong thing up here. So there's some pictures in Eberlin's book, Foresight. He's a spectrophotometer, and he took pictures of them. And you can just see those cells. They just distribute the light to the different cells. And so that actually increases our ability to see. It's not, and so now it's a perfect eye. We have a perfect eye. And we still have a blind spot. Does that affect anybody in here? No, it doesn't affect us hardly at all. So anytime you see a purposeful arrangement of parts, that means design. That's design. If you have to have several things working together, and if you take one apart and it doesn't work, then that means that was designed. Evolution can't work that way. Evolution only works mutation at a time, slow, gradual mutations over time. And they think they accumulate. But how can an organism live if it's not working? It just doesn't work. So anytime you see a purposeful arrangement of parts, that indicates design. And that's what I really wanted you to see. I was in Lubbock. I saw in the paper, one of the tech professors, an engineer, was talking about scientific and engineering evidence for creation. And I'm thinking, whoa, that's interesting. So I went and heard him. Heard his talk, wonderful man. Afterwards, we talked a lot, and he said, do you want my slides? And I said, I sure do. And so he gave me his slides so I could show you this. It's just really kind of fun. He looked at the peacock feather. Actually, it wasn't him. He just read about this in the National Academy of Sciences that these Chinese researchers were working on. And it's talking about the peacock feather there. So he calls it bio-nanophotonics because he's dealing on a really tiny level. So that's what your eye sees when you look at a peacock feather. But there's absolutely no colored pigments in that feather. None. No colored pigments at all. If you look at a magnification of that, that's what you're going to see. when you look at it. And so that line right there is 0.0005 millimeters. So tiny, tiny, tiny. You see these photonic lattices in here. Those photonic lattices work like this. If that's a photonic lattice there in the middle, when light strikes it, it's going to absorb all the colors, but let one color come through. Do y'all see the red color come through? Yeah, now watch when I change that photonic lattice. A different color comes through. So the eye on a peacock is all these different arrangements of these photonic lattices letting different colors of light come through that you can see, and it gives the picture of an eye. Is that not incredible? So he was showing the scientific study from the people in China, and this is from the proceedings of the National Academy of Sciences. Look at what they say there, right there. They say coloration strategies in the peacock feathers are very ingenious. That's a design term. That's an intelligent term. That's a very ingenious thing. So they're starting to see a lot in science that looks like it's designed. This is the Madagascar orchid. I really like the orchid. Down here, it has this spur on it that comes out of the back of the flower, comes all the way down, and right there is a nectar that's just almost irresistible to any moth. So who can get that nectar? This guy can. It's the hawk moth. He has got a proboscis there that's 16 inches long. So he can stick that thing down in that flower, go way down, and get that nectar in the bottom of it. Is that not incredible? And so there you can see him going in, and there he's dusting pollen on his face. And so when he pulls out, he's got pollen on his face. He goes to another flower. That's the only way this flower is pollinated. Is that not incredible? That is just so fun. It's fun. Here's another flower. This is the manuka flower, and a bat pollinates it. So you can see a flower right there. You can see a flower right there, a little flower right there. This flower is open. So what happens is on each flower, there's these two little discs. You can see them like there. And those two little discs on the flower, when it comes time that that flower is mature and it's ready to open, those discs kind of form a cup, and the bat can echolocate off that cup. And right between the two discs is a slit, and the bat knows to, as he's coming in, to stick his tongue in that slit, and he's going to get a lot of good nectar when he does that. And what happens is, when he hits that flower, there you go. He came from this direction. He hits that flower, sticks his tongue in that slit, and it triggers a switch, and it drops all the flower parts down, the stamens and the pistils. Well, he swings around as soon as he hits, and as soon as his rear end comes up, it dusts pollen on his rear end. And then he goes to the next flower. And when he goes to the next flower, he only spends 0.2 seconds per flower. When he goes to the next flower, he hits it from this direction. It swings his rear end around. That triggered a switch. It drops the pistil down, and it pollinates the pistil. That's the only way this flower is pollinated. How do you get a plant and an animal with just the right parts to make it work and be just the right size to make pollination work? Had to be an intelligent designer. This is my last one, then we'll be through for the day. It's only 1:33. You are going to be just fine. This is an orchid. This is an orchid in Australia. And that's what it looks like. And so you'll find this orchid down in Australia. You'll also find this wasp. It's the thynid wasp. But you only see the male. You will only see a male thynid wasp. The female lives underground. She only comes out one time a year. What she does is she comes out of the ground, climbs up a plant, releases a pheromone to attract the male. The male comes and mates with her. Then she goes back down in the ground, lays her eggs, and never comes out until the next year. So that's the story. Well, let me go back. This is so fun. There's the orchid. This orchid, at the time that that female butterfly or moth is supposed to come out, the orchid releases the same pheromone. The orchid does. And it's got this structure that looks like the female of that moth. So it releases the same pheromone. There's that, in the plant world, we call that a dummy female. The male comes and tries to mate with that dummy female. That is a hinge. And when he tries to mate with that, it brings him around and dusts pollen on his back. It looks like that. It brings him around and it dusts the pollen off the flower onto his back. That is the only way this orchid is pollinated. Now tell me something. How do they get an orchid that has that structure on it that looks like the female and has a hinge on it that has just the right torque, that if a male attaches to it, it brings him around? And do y'all get my point? That had to be designed. Those are design features. I think that's what God is trying to get us to see. If you just look at his creation, you can see his eternal power and divine nature in what has been made. We see in the unique mechanisms of sexual reproduction, purposeful arrangement of parts, irreducibly complex systems, all are indicators of design. This is my favorite verse. I ought to have you memorize it before you come back tomorrow. Romans 1. For what can be seen or what can be known about God is plain to them, because God has shown it to them. Ever since the creation of the world, his eternal power and divine nature, invisible though they are, have been understood and seen through the things he has made. Can I read that backwards? Start at the bottom. Through the things that God has made, you can understand and see, invisible though they are, his eternal power and divine nature. Ever since the creation of the world, they've been seen because God showed it to them. God made it plain to everyone to see. Just look at what he's done in nature and you can see his power and all that. Okay, here's where I go for a lot of my information. This is my last slide. Evolutionnews.org is the intelligent design group. It's no longer called evolution.org, although if you put evolutionnews.org, it'll take you there. It's now called scienceandculture.com. They are the intelligent design people. They work with things that look like they're designed. They work with DNA, really good resource. And they change it every day. They have different topics every day. So you'll really like it. This is the same group, discovery.org. And so that's the Intelligent Design main website. It's a really good place to be. Now, they don't mess with the age of the earth. To them, the earth could be millions of years old, it doesn't matter, but they really focus on the design part, which is really, really good. In the Church of Christ, David Reeves, just look up davidreevesministries.org. I think he's right outside of Nashville. Really good. He does a really good job of bringing things about creation out. Of course, Apologetics Press has been around for a long time. Kyle Butt and Eric Lyons, and there's several people that work in there that are really good and talk about evolution and creation. So those are really good resources. ICR is good, Institute of Creation Research, ICR in Dallas. I used to not like them, but now they're doing a lot of science research. And so I really like Timothy Clary, who looked at all of the oil well rig drillings and did all the different mega sequences. He's with ICR, and so he did a really good job with all of that. Okay, it's only 1:38. Are we okay? I'm okay. All right. Okay, Brian, thank you. Yeah, thank you.
Will you pray with me?